Welcome back, folks. The last time we'll be talking about the science of how drugs work while we're together. So let's learn about the science of sodium pentothal and how it works. So let's first just take a look at sodium pentothal for a moment and talk about it. So here is a structure of sodium pentothal. We have this kind of ring and this little tail looking thing. So a structure that looks like this means it's lipid soluble, or it would dissolve in oil. And what this means is that when it enters our body, it can get into the brain very quickly. And so this explains why it kind of works so fast. As chemists were trying to find an anesthesia that would work, faster than 20 minutes. A molecule like this is excellent because as soon as it reaches the brain, it can start to work. Also, this drug is able to, once it reaches the brain, kind of rapidly spread out through the entire body. And so as its concentration then decreases in the brain, this describes its short acting times, why it really only works from 3 to 5 minutes or so for very short surgeries. So how does it work? Well, in order to understand how sodium pentothal works, let's first talk about what is going on in the brain that allows it to work. So I have to introduce you to this class of proteins called GABA receptors. Specifically, GABA A receptors. So this is a protein. I've shown you its picture here, GABA. It's a protein that's made up of these 5 kind of different parts that sit in the membrane. And they're called GABA receptors because they bind to gamma aminobutyric acid, which for short is just called GABA. GABA is an inhibitor, right? And if we're remembering what that means, inhibitor means turns off. And these proteins act as channels, so they kind of let things go through them. The fancy name is a ligand-gated ion channel, but we will talk about what that means in a second. Finally, on this slide, folks, I want to point out to you what GABA looks like. It is this molecule here. Okay, so how does GABA work? So the molecule GABA comes in and binds to or is grabbed by the protein GABA receptor. So here we are in the brain. Here's the cell membrane. Outside of the cell, we call the extracellular space, and inside of the cell, remember, the liquidy interior is called the cytosol. And when GABA binds onto the protein, what happens next is it kind of opens up to let this molecule, chloride ion, chloride like table salt, through. So that chloride ion comes in and goes all the way through the GABA receptor to the inside of the cell. And then GABA can leave. So that's how this works. And this receptor, recall, on the previous slide, I said acts as an inhibitor. So this GABA binding and allowing chloride through is turning something off. What exactly is being turned off? To understand that, let's remind ourselves a little bit of what we learned about how the brain works. So last time we were together, I kind of talked us through this picture of a nerve cell when we were learning about how Adderall works. So recall that here is where the neuron starts. So a signal starts here, and the signal is sent through the neuron or the nerve cell, and we call that the action potential. The action potential is an electrical signal, and eventually that electrical signal reaches the end, and so neurotransmitters are released, and this is a chemical signal that can then go on to the next cell and tell it, hey, keep sending the signal, keep doing the communications, right? But last time we were together, we didn't really go into any detail about what an action potential is. So what is an action potential? So nerve cells or neurons normally have a charge difference across their membrane. What I mean by that is if you zoomed into a neuron, you would see that on one side of the membrane, it has a positive charge, and on the other side of the membrane, it has a negative charge. And this is just how it is, just kind of at rest. We have a positive charge on one side of the cell and a negative charge directly on the other side, on the inside of the cell. And an action potential occurs when this charge difference changes. So I'm going to show you how this works in this fun GIF. So here is an action potential happening inside of a cell. You can see that we start off with a positive on one side and the negative on another side. But this action potential happens when all of a sudden on the outside, the negative flips. And that negative charge traveling down the nerve cell is what we call an action potential. That negative charge traveling down the nerve cell is the signal. Oh, sorry about that. Pen got turned off. And the action potential is the electrical signal. So when this neuron is trying to send a message, it causes this action potential to happen. When these GABA receptors work, and when chloride ion goes through the protein, what's happening is we have a sedative effect because that negative charge on the chloride ion going inside prevents the action potential from happening. And I'll show you on the next slide exactly what I mean by that. And this is why GABA is an inhibitor. Sodium pentothal, on the other hand, works exactly the same as GABA, okay? So GABA comes and binds up top and allows the chloride through. Sodium pentothal, it binds somewhere else. It binds here on the bottom, but when it comes and binds, it also lets chloride through. Why is letting chloride through acting as an inhibitor? So recall, for an action potential to work, we need the charges to flip. We need the outside to become negative, and we need the inside to become positive along the neuron, just like this on the top. But if we send chloride through, we're sending a negative charge to the inside. So if this negative charge is being sent to the inside, we're not going to have this positive charge traveling down on the action potential. If the negative charge is on the inside, we're not going to have a negative charge traveling along the outside of the neuron in the action potential like we're seeing up here. So what happens is after GABA or after sodium pentothal, there is no action potential. It was inhibited, it was blocked, all because we have now this negative charge being moved from the outside to the inside. And you can imagine that the action potential of neurons, if it's blocked, it should cause the anesthetic effect, because brain signaling is effectively blocked or decreased. So that is how sodium pentothal works as an anesthetic. It goes to the brain, it works on its receptor called GABA A receptor, which blocks your brain from sending signals. Of course, there are some side effects of sodium pentothal, some of which are related to its use in lethal injection. So sodium pentothal can lead to respiratory depression. If your brain kind of slows down, it'll slow down all of its functions, and your brain is responsible for keeping you breathing. Sodium pentothal can lead to withdrawal effects, and it can also lead to liver injury. Recall to when I introduced that the liver is the primary place where drugs are metabolized. So it can kind of mess with the metabolism of stuff in the liver, potentially leading to injury. And that wraps up how sodium pentothal works. I'll see you in class, and I'll leave you with this cat meme of a cat taking a little nap.